It has been 37 years since HIV was first discovered as the cause of AIDS, but there is still no vaccine available against this disease. This contrasts with the rapid speed of the COVID-19 vaccine development and distribution. According to a professor of pathology, the delay is caused by the remarkable HIV strain diversity and the immune evasion strategies of the virus. In spite of the challenges, scientists are still researching a variety of solutions and have already made progress in the fighting against this complex virus. Smallpox has been eradicated from the face of the earth following a highly effective worldwide vaccination campaign. Paralytic poliomyelitis is no longer a problem in the US because of development and use of effective vaccines against the poliovirus. In current times, millions of lives have been saved because of rapid development of effective vaccines against COVID-19. And yet, it has been 37 years since HIV was discovered as the cause of AIDS, and there is no vaccine. Let me describe the difficulties in the development of an effective vaccine against HIV/AIDS. Vaccines have unquestionably been society's most potent weapon against viral diseases of medical importance. When the new disease AIDS busted into the scene in the early 1980s and the virus that caused it was discovered in 1983 to 84, it was only natural to think that the research community would be able to develop a vaccine for it. At a now famous press conference in 1984 announcing HIV as the cause of AIDS, then U.S. Secretary of Health and Human Services Margaret Heckler predicted that a vaccine would be available in 2 years. Well, it is now 37 years later and there is no vaccine. The rapidity of COVID-19 vaccine development and distribution puts the lack of an HIV vaccine in stark contrast. The problem is not failure of government. The problem is not lack of spending. The difficulty lies in the HIV virus itself. In particular, this includes the remarkable HIV strain diversity and the immune evasion strategies of the virus. So, should the researchers give up? No, they shouldn't. One approach researchers are trying in animal models in a couple of laboratories is to use herpes virus as vectors to deliver the AIDS virus proteins. The herpes virus family is of the persistent category. Once infected with a herpes virus, you are infected for life. And immune responses persist not just as memory, but in a continually active fashion. Success of this approach, however, will still depend on figuring out how to elicit that breadth of immune responses that will allow coverage against the vast complex of HIV sequences circulating in the population. Another approach is to go after protective immunity from a different angle. Although the vast majority of HIV-infected individuals make antibodies with weak, strain-specific neutralizing activity, some rare individuals do make antibodies with potent neutralizing activity against a broad range of HIV isolates. These antibodies are rare and highly unusual, but scientists do have them in their possession. Also, scientists have recently figured out a way to achieve protective levels of these antibodies for life from a single administration. For life, this delivery depends on a viral vector, a vector called adeno-associated virus. When the vector is administered to muscle, muscle cells become factories that continuously produce the potent broadly neutralizing antibodies. Researchers have recently documented continuous production for 6.5 years in a monkey. So if you've been asking yourself why is it so much harder to develop an HIV vaccine than a COVID vaccine, the answer is even though they are both viruses, they are totally different. Problem is the biological properties that HIV has involved make development of a successful vaccine very, very difficult. What are those properties? First and foremost is the continuous and unrelenting virus replication. Once HIV gets its foot in the door, it's gotcha. Many vaccines do not protect absolutely against the acquisition of an infection, but they are able to severely limit the replication of the virus and any illness that might result. For a vaccine to be effective against HIV, it will likely need to provide an absolute sterilization barrier and not just limit viral replication. HIV has evolved an ability to generate and to tolerate many mutations in its genetic information. The consequence of this is an enormous amount of variation among strains of the virus, not only from one individual to another, but even within a single individual. Let's use influenza for a comparison. Everyone knows that people need to get revaccinated against influenza virus each season because of season-to-season variability in the influenza strain that is circulating. Well, the variability of HIV within a single infected individual exceeds the entire worldwide sequence variability in the influenza virus during an entire season. What are we going to put into a vaccine to cover this extent of strain variability? HIV has also evolved an incredible ability to shield itself from recognition by antibodies. Enveloped viruses such as coronaviruses and herpes viruses encode a structure on their surface that each virus uses to gain entry into a cell. This structure is called a glycoprotein, meaning that it is composed of both sugars and protein. But the HIV envelope glycoprotein is extreme. It is the most heavily sugared protein of all viruses in all 22 families. More than half the weight is sugar, and the virus has figured out a way, meaning the virus has evolved by natural selection to use these sugars as shield to protect itself from recognition by antibodies that the infected host is trying to make. The host cell adds these sugars and then views them as self. But have researchers given up? No, they haven't. Well, that's all I have for you today. Let's hope that one day we get the HIV vaccine too. My name is Rose and see you in the next video. Bye for now.